So, welcome to the Curation and Biodiversity Informatics course, the first week. Natural History Collections, the fourth of these presentations. And today, this is going to be an example from start to finish of a marine survey and how to build up a collection at the museum, how to use that and how to connect that to the old collections and the data involved in it. So, next slide, please. Here we go. In the early 2000s, Sweden was the first country to begin with red list assessment of marine invertebrates performed by the Swedish Species Information Center. And there was an evident lack of knowledge on present occurrence, distribution status, and trends of the species. And the majority of these are found at the west coast. So, the Swedish Taxonomy initiative, which is part of the Swedish Species Information Center, they decided on a survey along the entire west coast with focus on Bantix communities in offshore areas where they have the largest data deficiency. So what you see here on these pictures below to the left, shows the bottom at Skagerrak and Kattegat using the OLEX bathymetric system. You see every time a trawler goes, you get new data. So it's heavily trawl area and heavily affected by fishing in that sense. And to the right, you have a multi-beam scanning sonar picture showing the Koster fjord trench in deep purple, and what's outside the Koster islands, which is going to be investigated in this study. So the next slide, please. Yeah. So it's called Svenska Artprojektets Marina Inventering, Samarin, 2006-2009. That is the marine survey conducted by the Swedish Taxonomy initiative. Next slide. So there was to be a lot of preliminary planning, and the museum was involved. The research vessels chosen, there was Skagerrak 2, Arne Tiselius, and for the last year a Norwegian boat. The OLEX system for coordinates and mapping the seafloor. And establishing of the routines for the workflow on board to get the most information out, the most use of it afterwards, so it should be open. There was about 370 stations chosen, of which 70 was visits of old Jägersköld stations from the 20s and 30s. I'll return to that later. And the sampling methods chosen after bottom type, and with the type of field protocol to fill in efficiently and to get all the data you needed to trace the locality, the depth and the coordinates, the species, the determiner, everything. And direct estimation of number of individuals or common species in each sample. And some difficult materials studied on board directly by a specialist at the boat. And then how to collect reference material of each species and do fixation, conservation and preliminary labeling on board. And have people keep in check of this. For 2009 also collaboration with the Norwegian Bioskog project. And to the right you see pictures of the stations for each year from 2006, 2007, 2008, 2009. You see there's quite a lot of places. Yeah, next slide. So for the different bottom you need different sledges and dredges that you drag after the boat. From rock, hard rock, to the softest clay. And a lot of the bottom, especially in the Skagerrak, is very soft mud. Okay, you don't have to, you just want to have a bit of the bottom where they have the most of the animals. Okay, and then next slide. So this shows some of the work on board, not in the lab, but outside. And there's a lot of clay in the sea. And it's been sieved all the time sieve to get the animals out. To the left you see Mattias Hobbs sieving away with a bun in his hand. And in the middle you see Anna Karlsson, who is the project leader. And to the right you see a Varén sledge to use for soft bottoms. And in the blue raincoat is Mats Berggren, in the middle there. That was the project leader from the practical side. Next slide. At the museum, material that was sampled and transferred to the museum, there was for sorting, in cases further sending out to specialists for determination, digitalization of data from field protocols, check of species list from determiners, coordinates and other locality data. And then we also made a change from the preliminary field containers, changed of ethanol, sometimes formalin, to vessels for long time storage. It's stored finally in ethanol, mostly. And the final labeling also. So in total 9,000 collection lots and 25,000 observations in total in the database. And 40% of the collected species was identified afterwards in the lab, and 60% were identified in the field on the boat. And there was many new species for the country, and found even some that was new for science. Next slide here, please. So some of the results of this, we got substantially improved knowledge base for red list assessment. That comes out every 50 years, a new red list for Sweden for animals and plants and fungi. And you got better knowledge for marine management, as for the identification of areas with high biodiversity. So if you go to Artdatabanken, the Swedish Species Information Center, you can download the final report for the submarine survey. You see the link there. And also the data was handed over to the County Ministry Board and the Swedish Environmental Protection Agency that used them for the marine management. Can I have the next slide? So what we got in newly protected marine areas in the last years. There was 2 large new Natura 2000 areas in the county of Skåne that was decided on by the government in December 2016. The marine area outside Kullaberg and Höganäs are becoming the nature reserve called Skånska Kattegatt in the southern Kattegatt. And the European Union Commission in January 2017 decided on fishing-free zones at the area Bratten area by suggestion of Sweden, that is outside of Smögen. A few further nature reserves are planned in the regions of Västra Götaland and Halland. And you see to the far right along the whole coast, the protected areas, and in the middle you see the Bratten area and different restrictions at different areas in that. The Bratten area is it's like ravines and cracks and crevices on the seafloor, very, very varied and high diversity. So, next slide please. But meanwhile, while the boat was out doing the survey and collecting, at the museum we performed the digitization of another older survey, so actually done by museum director Leonard Axel Jägerskiöld. You see him there to the right. He conducted a survey of Bantix invertebrates at the Swedish west coast from 1921 to 1938. And that is the only previous survey undertaken along the entire west coast for reference. So a large reference collection from the survey was stored at the museum in Gothenburg with over 34,000 lots determined by about 30 specialists, just as similar to submarine. But everything is now, there was nothing before, is now digitized in specified data system and available through portals like GBIF or the Species Portal. Not only that, all associated collection equipment and the documentations like field notes and so on have been stored at the museum and could be used. So we can have the next slide. So this marine Bantix survey was 450 stations along the entire coast from Svinesund in the north, close to the Norwegian border, to the Kullaberg in the south, from the shallow beaches to the deep offshore trenches. Next slide. So this is what it could look like in the 30s. A lot was very different, but these dredges and sledges largely were the same actually, and the sieving aboard the ship. Next slide. So new coordinate estimations were done of the Jägerskiöld stations, because many of them was previously only bearings. But now you see, you can carefully examine depth and locality and the species composition to know exactly where they were, so we can do a revisit of 70 of these. So, and then we have an estimation of the effort of the sample bottom surface area. So you can see, look at the equipment and the data for notebox, how many times they've done this. So we could get a statistically comparable results on the old and the new surveys actually. So you can do an analysis of the two data sets from this survey and to submarine, so that you reveal large scale changes as presented in a paper from 2018. You can see here, Obstedal and me and some other people, but more on that later in the course, how that was done. To the lower right here, you see that all the collections, localities at Jägerskiöld and those at submarine survey. Yes, next slide. So, but now that we had the data to go on with, but we also had the collection, the material. So it could be used further. So as a final step in this long process, the newly collected and identified material from specialists that was used to establish a reference database of barcodes, CO1, for marine invertebrates. In a joint Norwegian and species project, we had a Norbol and a Swebol together with the eyeball barcode of life project. Yes, so different phyla of marine invertebrates. So all data from those are available on bold systems now, with links to voucher specimens in the collections both in Sweden and in Norway. Yes, and then it's now it's free for further research on the material. Yes. So, and now for the very final slide. Yeah. Thanks for watching, and see you again. Goodbye.